We're still learning, in fact, what there is to be, that the virus is showing us. If we're just smart enough to see what this virus is trying to teach us. Somebody once told me viruses never make anything up. And they're actually fairly advanced evolutionarily. And HIV is particularly clever in that it has taken a way of turning on its genes, turning them on and off, that's like very efficient and very simple. And it involves it actually is constantly making little pieces of RNA of its own gene, and it's but then it makes this little piece of RNA and then it stops. The RNA polymerase, which is sitting on the DNA and making the RNA, stops and falls off and recycles round and round and round. And the virus, that's how it maintains a latent state. And so it's sort of dormant, but it's still activated. It doesn't need much to get it going. Everything is assembled and ready to go. It's just that last little piece that keeps the RNA polymerase chugging along instead of falling off. And
what last little piece?
The last little piece is the activation of the cell that's been infected by the virus. And the cell itself will make proteins that are normal to an activated cell, and those proteins will then go and activate the HIV promoter region as well. So the promoter of an HIV is actually just like one that we have, and in fact the areas that will bind these activating proteins are areas, they belong to any activated gene. And the virus just sort of is mimicking them, and the way that when it's still just latent and quiet, it again is using all the same things that our normal host cells have. For HIV, it's the inflammatory cells, the T cells in our immune system. And it just, what it does is it makes a little fragment of RNA that forms a stem in a loop. And it's like 60 nucleotides long. It's very small, little stem loop structure that you can actually detect in cells that are infected, and it's a sign of latency. When every once in a while, you get a full RNA made, a full viral RNA made, and a virus, the viral protein called TAT is made, and it's a protein that will then go and interact with this stem loop. But it's too slow a mechanism. The virus is basically sleeping, but as soon as you activate that cell, and the virus starts making more and more of its RNA, it starts making more and more of this TAT protein, its own special protein. And that protein comes along and attaches to this stem loop. The protein attaches to the RNA stem loop and attracts in the final protein that's needed to get the RNA polymerase to just get rolling. So at that point, very efficiently, the system basically takes over the cell. You start making tons and tons of virus. So what the virus sort of first really demonstrated clearly for us was that you could have a very small RNA stem loop that could be like a little molecular switch that could, once there was enough of this viral TAT protein recruiting in the final step that would trigger the RNA polymerase 2, and when we did the film, it's that moment where the little light flashes and the C-terminal domain of this big RNA polymerase protein is triggered, and off it goes. So at the time that was first discovered, there was no example of that going on in any kind of other gene, and it was considered to be a fluke of HIV and something unique. And that was around, I don't know, maybe, this must be going on about 30 years ago now, 25, 30 years ago, they first started to clarify what was going on with HIV. And it seemed kind of crazy that it would be a fluke, because it's so efficient. It's such a great way of turning a gene on, especially those genes you need very quickly to be turned on, because you already have everything in place, and you're making these, HIV is making these little RNA fragments, and the RNA fragments are going out and acting like little flags, in fact, saying, here I am, here's the HIV genes, here I am, like a little radar. And again, and that's sort of the idea that I got for our radar for the film.
What in our bodies, in our systems, might be needed to be turned on quickly like that? What's the analogy?
It turns out that there's a whole group of proteins called quick response or stress response genes, and they get triggered when the cell is stressed by heat. Again, heat and ionic changes, those things are very stressful to a very basic cell type. And if a cell is sensing that there's something awry, it will turn on these genes. And there's actually a whole series called heat shock proteins that get turned on. And these are the kinds of genes that get turned on when a cell is dealing with stress. So for different cells, that's different things. But there's stress response genes, and they can be activated very quickly. And it turns out, if you look in the cell, that you can find these little transcripts that are, flagging the presence of those genes, always present in the cell. And then the more they looked, the more and more genes they found that were making these little transcripts. And currently they now know that almost all genes make little transcripts, not only in one direction, but in the opposite direction as well. And nobody knows what those are doing.
So how did those findings about HIV and virology affect your personal understanding? How did it modify your thinking about RNA?
Yeah. It was actually, I was already thinking it's impossible that mammalian cells aren't using this system. And my first research work was done on trying to figure out how transcription works. How is RNA made? What turns things on, and then what turns them off? And so basically, once they discovered these small RNA fragments that could guide bigger proteins around, then it became sort of easy to imagine you would have a gene sending out these little radar fragments, these little flags saying, here I am, I'm a specific gene. And some other gene, maybe that is going to be interacting at some level, that these two genes are making little fragments that actually match. They're complementary matches. And now they can find each other because you have these Argonaut proteins. So all of a sudden you could have one gene very simply contacting another, just based on these little RNA pieces, and then the RNA that gene is just making as flags. So boom, suddenly you have two genes that can connect. Well, once you have two genes connecting, well, why not three? Why not four? And that was the beginning of the idea it could network.
So is this a system that, not that our cells are the very first version of our cells way back in time got from virology, or that virology got from
I think about that.
System?
Most people think that it's the viruses came first, and then somehow the viruses infected the cells and got the thing rolling, but that just doesn't make sense to me. It makes much more sense that you have some very basic RNA system in place that genes are using to communicate in the most basic way, and then that system is co-opted. And a virus is nothing more than some RNA or DNA packaged in its own little proteins and enveloped in such a way as it can sort of jump from person to person. But the very basic mechanism of making a piece of RNA from a piece of DNA, and then in reverse, that probably came first. That's too elegant a system for it to be something that evolved later and separately in a virus.
Mm-hmm. So maybe the difference between what we think of as virus and non-virus is sort of an arbitrary line.
It's arbitrary. And then you can take that closer and closer to, a more basic mechanism. Again, that viruses are doing. Viruses jump. Well, turns out RNA can also jump itself in the nucleus. And that's part of evolution. That's probably what triggered this huge burst of evolution that started unfolding at a very fast rate around 450 million years ago.